This is my third lecture on the origins of life on Earth, from molecules to cells. We're going to be looking at some hypotheses around how some of the first molecules and cells might have formed, looking at prebiotic chemistry, or chemistry before life, and abiogenesis, which is the generation of life from non-life. And in order for us to study this, we need to look at recreating early Earth somehow, or look at early Earth analogs, or in silico modeling, molecular modeling of some of the molecules that may have existed in early Earth. Some of the great early Earth analogs that scientists use today are hydrothermal vents and modern-day stromatolites, which occur in a couple areas, such as Shark Bay, Australia, as well as hot springs, where there's a lot of heat and a lot of minerals and a lot of organic molecules. We also use in silico recreation to reconstruct some of the conditions that may have been there and use some modeling. So we're going to look at different aspects of this in terms of the hypotheses. One of the first experiments to recreate early Earth in vitro, as in a test tube in the lab, was the Miller-Urey experiment in 1952. And they experimentally recreated an early Earth environment using an atmosphere of water vapor, methane, ammonium, and hydrogen with an electric spark to mimic lightning. And they generated actually several amino acids this way. And this was all abiotically, without life, and spontaneously. So this wasn't a chemical experiment where they tried to reconstruct an amino acid. They wanted to see what could form spontaneously given very minimal conditions. And this really paved the way for further experiments where scientists would try to create different types of biological molecules, from amino acids, nucleotides, lipids, from really just bare essential molecules that were likely found in early Earth's atmosphere and environment. So an example of recreating early Earth in silico as well as in vitro is the systems chemistry approach. And this combines in silico modeling and predictions with the in vitro laboratory testing. And it looks at the chemical interactions that were possible in the early Earth environment, and then tests some of these hypotheses that are generated. And really, the more we learn about what was likely the environment on early Earth, it was pretty messy. There was many different molecules interacting in ways that don't necessarily happen in modern biology. And systems chemistry really attempts to account for this. So one of the very recent studies that looks at this is the role of sugar backbone heterogeneity and chimeras in the simultaneous emergence of RNA and DNA. So we're going to look at this. And this is actually built on a previous hypothesis called the RNA world. That RNA was the first biological molecule because it could form secondary structures and catalyze chemical reactions, much like proteins. But it was also self-replicating. So it had two really important aspects of what was important for biology. But when we look at them and model the interactions of RNA and being able to replicate another RNA, it seemed very unlikely thermodynamically. So this group actually looked at forming an RNA and DNA chimera. You can see the blue and yellow dots in the bottom frame formed together. And they found actually in vitro when they tried when that molecule replicated itself as RNA and DNA is capable of doing spontaneously, they created a pure RNA strand and a pure DNA strand. So this actually points to probably a more likely scenario, accounting for kind of the messy chemistry that was involved. So what systems chemistry really tries to do is to build on the idea that many 1, 2, and 3 carbon molecule precursors can be synthesized abiotically from methane, carbon dioxide, carbon monoxide, ammonium, and some nitrogen molecules with either the addition of a spark ultraviolet radiation, or shock waves, all of which would be present in early Earth from our current understanding of that time period. Many more complex molecules can then be assembled from that, again, spontaneously, abiotically, including the nucleic acids, nucleotides, and lipids. Systems chemistry really says that, and this touches on stuff from our first lecture, that life could emerge when coexisting molecules have the capacity for metabolism, or metabolic machinery, compartmentalization, and information replication and storage. So that would be metabolism, some type of protein or RNA or DNA are also capable of that, compartmentalization in terms of lipids, and information storage and replication in terms of DNA and RNA. Furthermore, so now we're going to see how lipids fit into this equation. Lipids and membranes. Compartmentalization is very much a key for life to have developed. In concentration, they serve to concentrate molecules together and increase reaction frequency. They would have also created compartments for different chemical environments such as pH or oxidation state to support that environments necessary for different reaction conditions within close proximity to each other. They also serve to create an energy gradient, such as protons or electrons concentrated on one side of the membrane that can then cross to the other, releasing energy in this manner, which is really the basis of of how life works today, to drive energy metabolism within cells as we know it. We also know that lipids, from recent research, that lipids can self-organize into membranes and even liposomes. So they can form these long membranes. Those can also bleb off to form little liposomes, little smaller structures that we talked about in our previous lecture, that can then contain some of the other biological molecules, such as RNA and DNA and some amino acids. And we can see how that chemistry within those liposomes can actually start to build something that looks like a modern-day cell. We know that phospholipids spontaneously can create nice membranous structures. And a recent paper that came out to kind of look at how this may have worked in early Earth and actually incorporating electron shuttle molecules that have been found in asteroids show the potential for chemoosmotic energy from primitive cellular life. Proton gradients are generated across the lipid membrane by redox reactions coupled to meteoric quinones. So membranes could separate the production of electrons on one side and the consumption on the other. And this would drive metabolic reactions. And the quinones that are found in some asteroids are capable of shuttling those electrons from one side of the membrane to another, where that electron can then be used in another reaction. And this is, again, a really key basis for how life works on Earth today. And when we look further, take another step back to looking at hydrothermal vents, which represent probably a very likely place for life to have started, and there's good evidence for that when we look in the fossil record. The reason that hydrothermal vents were likely a good environment for this was that the hydrogen produced through the serpentization reaction occurring in hydrothermal vents would have been ideal for chemical conditions to support the formation of biological molecules. Formate, and recent work done on hydrothermal vents over the last several years shows that there are pathways for synthesis of abiotic synthesis of many organic molecules, including formate, which is one of the key metabolites used in living cells today. So really, ancient hydrothermal vents were very likely a place for life to have begun, and that's supported both by modern chemistry as well as the fossil record. So in summary, to study the origins of life, we must find either early Earth analogs, recreate early Earth, or model it with computer programs. And the Miller-Urey experiment was one of the first to show that amino acid building blocks could be spontaneously made by abiotic systems under the right conditions. Systems chemistry approaches support a DNA-RNA hybrid molecule as a possible initial biological molecule. And systems chemistry really allows for the complexity of the initial early life chemistry. Furthermore, the role of lipids and membranes were essential for concentrating molecules and the compartmentalization and creating energy gradients. And that's it for today. Thank you.